So the overview of meiosis is that during that interphase is going to precede meiosis because this is a very important phase where all chromosomes are replicated. So during meiosis one, which I just showed you, those homologous pairs are going to separate from each other, reducing the number of chromosomes from the 2N to N. And that's why we refer to it as the reduction division of meiosis. And specifically, it's meiosis 1. During meiosis 2, those sister chromatids separate and each daughter cell gets equal share of chromosomes. So we call it the equatorial division of meiosis, which is just like mitosis. So we're going to go into a little bit more detail about meiosis here. Meiosis 1 is the reduction phase. You should know that by now. So the chromosomes are reduced from 2N to N, which is essentially 46 being reduced to 23. Remember, the first part of meiosis 1 is the most significant part, called prophase 1, and it has events that aren't in mitosis or meiosis 2. Some of those events include what's called synapsis and crossover or chiasmata. In synapsis, homologous chromosomes are going to pair up to form tetrads that consists of four chromatids. So it kind of be like this. We have one chromosome here, and then we have another chromosome that's going to pair up with it, which is now a tetrad. Then what's most important is there's going to be an exchange of genetic material between the male and the female chromatids. This results in the unique chromosomes that are mixtures of the maternal and the paternal chromosomes. So it mixes up the genetic material. So if we look at the end of this tetrad right here, there's an opportunity for this part of the sister chromatid to cross over with the other. And this is one of the reasons that there is the production of very diverse gametes. So all of these gametes are going to be very different in the chromosomes, the maternal and the paternal chromosomes that they actually have. Our next slide shows images that you have in your textbook, which are super, super helpful. So we're going to spend a few minutes going over these. It's kind of reviewing what we've talked about. So we can see the nuclear envelope shown right here, the chromatin. And what's going to happen is that nuclear envelope is going to start to break apart in prophase 1. And again, that prophase 1, I want you to remember that it's really the most significant phase because synapsis occurs. These homologous chromosomes come together to form these tetrads. And during synapsis, the arms of homologous chromosomes wrap around each other. It kind of would be like imagine yourself sitting next to your classmate and you're going to wrap your right arm around their left arm. And so, because of that, parts of your genetic material from your right arm can be shared with your partner's left arm, which represents genetic material as well. So it forms several crossover points, which are called chiasmata. So again, a tetrad is basically four chromosomes that are connected, which we can see in the image here on our left. So this is showing the tetrad. And in metaphase 1, the tetrads are now going to align randomly on the equator, which we also call the metaphase plate. During anaphase, then, the centromeres They don't separate during anaphase 1, but they form dyads, and those dyads are going to move towards the opposite pole of the cell. Finally, in telophase 1, which is kind of the opposite of prophase because the nuclear envelope is going to start to reform around the chromosomal material. So the two daughter cells, which are now haploid, enter a second interphase before meiosis 2 begins. So again, the most important part is to remember that meiosis 1 is the reduction division. On the right side of the slide, we're seeing kind of an overview of meiosis. So we go from 2N to 2N during interphase. So the chromosomes replicate, but during meiosis 1, we have the reduction division, where we go from diploid to haploid. And then meiosis 2, those sister chromatids separate, and each N is going to produce another N haploid cell. So we're going to relate that then to the names of the developing sperm or the developing egg. So we're going to use the term primary and secondary a lot when we talk about either spermatogenesis or oogenesis. The primary cell is always going to be diploid, so it has 46 chromosomes. And the secondary cell is always going to be haploid with 23 chromosomes. And that's going to occur during meiosis 1. So, for example, during spermatogenesis, the primary spermatogonium is diploid, but the secondary is haploid, and the same for the female. So keep that term primary and secondary in your mind as we go through the male and the female. Our next slide here is going to compare meiosis and mitosis. So for mitosis, we can see that we have 2N in prophase, and the result is daughter cells that are also 2N. For meiosis, we have the tetrad in prophase 1. We start with diploid, but end up with haploid at the end of meiosis 1 because that's the reduction division. And again, it's just like mitosis, the meiosis 2. So in DNA replication, that occurs during interphase. There's also interphase in meiosis. The synapsis, that does not occur in meiosis. It occurs in prophase 1, where the tetrads form and there's crossover of genetic material. The number of daughter cells for mitosis are 2, so each diploid produces diploid daughter cells. But in meiosis, there are 4 because there are 2 phases, and all 4 of them contain half the number of chromosomes as the parent cell. So the role of the body for mitosis is that it plays a role in development of multicellular adult from zygote. Tissue repair renewal ensures that there's constant genetic material of all the body cells. But in meiosis, it produces cells for reproduction, which are gametes. It introduces the genetic variability, which is super, super important. Reduces chromosomal number by half, so that during fertilization, the normal diploid chromosome zygote is going to have a diploid number of 46. So that will conclude our discussion of meiosis.